Good morning, everyone. Hi, my name is Ilana Berman, and I'm the ASLO Awards co-chair, together with Elizabeth Harvey, who unfortunately couldn't make it here today. So I'm really happy to be here all week and to present the great awardees that we will have throughout the week. So each year, the ASLO Association recognizes our colleagues for their outstanding achievements in the aquatic sciences. We are pleased to announce the winners of the 2024 ASLO Awards. The G. Evelyn Hutchinson Award was presented at the 2024 Ocean Sciences Meeting, and the Ramon Margalef Award for Excellence in Education has been presented to Bob Chen previously. The remaining 5 awards are presented here every day just before the plenary at the 2024 ASLO Summer Meeting. So we have a great lineup, and I hope to see you here every day to recognize their excellence. And behind choosing awardees is a lot of hard work of the award subcommittees. For each award, we have a subcommittee working to go through the nominations and to try and select every year. So thank you, all the members of the award subcommittees. And if there are any of you here today, please stand up so that we can clap for you and recognize your hard work. Anyone? And another important thing is for all of those looking forward to the subsequent years. The nomination cycle is opening right after this meeting. So please consider nominating your colleagues, your friends, your supervisors, anyone you think that is worthy of an ASLO award. These are very prestigious awards, and it's a really important process to keep recognizing our members' excellence. So today I would like to actually start with our first award for the week, and this is the Raymond Lindeman Award. The Lindeman Award honors a young author for an outstanding peer-reviewed English language paper in the aquatic sciences. And this year, the 2024 Lindeman Award will be presented to Dr. Richard LaBrie for his 2022 paper, Deep Ocean Microbial Communities Produce More Stable Dissolved Organic Matter Through the Succession of Rare Prokaryotes. This paper highlights an innovative experimental approach demonstrating that microbial communities strongly control the recalcitrance and fate of DOM, dissolved organic matter, with implications for carbon exchange between the oceans and the atmosphere. In the nomination letter, Professor James Kotner states that Lindeman's work was novel in that he was able to integrate fundamentals of chemistry, physics, and biology into what we now refer to as ecosystem science. Richard LaBrie is an early career scientist that exemplifies similar characteristics, and his recent paper on microbial interactions with dissolved organic matter is a beautiful testament to integrative sciences. Please help me in welcoming Richard LaBrie to receive the award. And Pat, can you come up and give the award?
So on behalf of myself and the ASLO board, the award subcommittees, and the award committee itself, it's my pleasure to present this award. And this award is really one of my favorites because it's looks like the classic limnology and oceanography cover. So, so I'm pleased to present this award to you,
Thank you.
and thank you so much.
I think we need to take a picture, the two of
Yes.
us.
And one final reminder, please remember, nominate your colleagues, nominate your associates, anyone can nominate. It doesn't have to be someone special to nominate. You all can be nominators.
We need a picture together.
So hi everyone, I'm Richard. I'm currently a postdoctoral researcher at McGill University, and it's an honor to be here today receiving this Lindeman Award for work that I've done during my PhD at Université de Montréal. So I'm here today receiving this award, but I want to acknowledge the contribution of all my co-authors that worked with me on that paper, and of my PhD supervisor in particular, Roxane Maranger. Thank you. This was a hard and a long paper to publish, but it was well worth it. I also want to mention that this study was inspired by a talk that Craig Carson gave back in 2015 when he himself received the Hutchinson Award for his work on the microbial relationships with DOM in the Mesopelagic Ocean. So why would we care about DOM and microbes in the ocean? Well, DOM is a huge reservoir of carbon, similar to the carbon in the atmosphere. And one question that intrigues me is, why is this pool of carbon not consumed by microbes, bacteria and archaea? Over the past thousands of years, it has accumulated to these large quantities. And there are currently two main hypotheses to explain this. The first one is almost 60 years old. It's the dilution hypothesis. It states that DOM consists of hundreds of thousands of molecules that are so diluted that they cannot be used up by microbes. The other hypothesis, also about 60 years old, is the recalcitrance hypothesis. And this one states that it's chemical properties that makes DOM inherently hard to consume by microbes. And this has recently been repackaged into the microbial carbon pump. But what exactly is this microbial carbon pump? In the sunlit portion of the oceans, we have this classic microbial food web. Phytoplankton release DOC, bioavailable compound as labile and semi-labile DOC that will be used up by microbes. But this process is not perfect. They will release or transform part of this organic carbon into more refractory molecules, molecules that can persist in the environment for hundreds years old and more. And it's this link, this production of refractory organic compounds, that is called the microbial carbon pump. And in this study, we wonder if microbes from the surface ocean, the mesopelagic and the bathypelagic ocean, were all equally able to make these recalcitrant molecules. And the idea behind this is that microbes in the deep ocean live in much harsher conditions than in the surface ocean. They seldom have labile and semi-labile carbon, and so they must use carbon that is inherently more recalcitrant, and so they could maybe use a wider array of compounds. And to test this hypothesis, we worked in the Labrador Sea. It's a subarctic region between Canada and Greenland. It's a unique environment in the ocean where the atmosphere will be in contact with the deep ocean. In the winter, there's a large winter convection that will bring surface water to the deep ocean. And it's so critical that for CO2 entrainment and oxygen entrainment to the deep ocean, that a whole NSERC program called VITALS, Ventilation, Interaction, Transports, and Transports across the Labrador Sea, was designed to study this region. And in this program, we were interested in the fate of DOC, because during this convection, organic carbon would also be naturally to the meso and bathypelagic ocean. But how does this convection work? So here in color, we have the temperatures, depth on the y-axis, and time on the x-axis. So in May, the temperature is high, the water column is stable. But when the winter starts in January, it breaks this top stratification. And in February, it completely breaks this stratification and the convection begins. This is a fast process that can reach up to 2.5 kilometers deep. In May, the temperature rises again and the surface is stable. So this represents one cycle of deep winter convection. When we put all of these convection cycles one after the other, we have a picture that looks like this. And naturally, we will have the depth of this convection that will change among years, sometimes reaching only in the mesopelagic, like in 2010, and sometimes reaching well into the bathypelagic ocean. So in this context of the winter convection, we place the study to see where the MCP is best enabled. So we sampled along the AR7W line. It's an oceanographic line that has been monitored for the past 30 years by scientists at the Bedford Institute of Oceanography. And in 2016, it was a special year. On the right, we see plots of density and SF6, two tracers used in physical oceanography. And we see a large, deep mesoscale eddy that is pumping water from the water mass below up to the newly ventilated Labrador Seawater 2060. And using different tracers, we calculated that at 1,500 meters deep, where we sampled for our bathypelagic samples, the water mass was composed of 80% of the newly ventilated Labrador Seawater and 20% water from the water mass below. And we believe that mesoscale eddies like this one may play an important role in the microbial carbon pump. I'll explain this a bit later. We could not go there during winter. It's too dangerous. So we had to emulate that water mixing using bottle incubations. So we use 8 liters of filtered surface water as the DOM being dragged down to the deep ocean. And then we added an inoculum, microbial communities, either from the surface to have a control, from 500 meters deep as a shallow mixing experiment, and from 1,500 meters, the depth of the convection in 2016. And we kept these bottles in the dark and at 4 degrees Celsius to mimic in situ conditions, and we sampled them for 18 months. These batch culture experiments are a really good way and an easy way to measure the amount of bioavailable DOC and their kinetics of degradation. So how much carbon is being consumed and how fast it is consumed by the different microorganisms that live there. But we wanted to go a bit further, and we opened these 2 black boxes to look at the composition of the organic matter and the composition of the microbial communities. Here I will focus only on the DOC, the DOM composition using high resolution mass spec and sequencing data, and how these interact in the microbial carbon pump framework. So to start with, DOC degradation over time. Here we see in green the surface, in light blue the mesopelagic, and in dark blue the bathypelagic treatments. They all reach the same value of about 52 micromolar, which is the background DOC concentration in the North Atlantic Ocean. But we see that the bathypelagic got there slightly faster, and they could be more efficient at degrading DOC. So we don't have a quantitative difference in how they consume carbon, but we can still see a qualitative difference. How is the DOM pool modified after these transformations? To look at this, we used FTIC-RMS. So this is one sample, and each point represents one molecular formula that is represented using its oxygen to carbon and hydrogen to carbon ratios. To summarize this information, we often use groups of compounds like aliphatics, unsaturated, polyphenols, condensed aromatics. And we see which groups are dynamics, either among lakes, along a river, or in our case, over time. So here we have the surface on the left, the mesopelagic in the middle, and the bathypelagic treatment on the right. I'll bring your attention on the red and yellow bars. So we see in the surface there's barely any variations after 3 months of incubations. But if we look at the deep treatments in the middle and the right, we see that the high oxygen molecules are being produced and the low oxygen molecules are being consumed. And so we already see that there are differences between the surface and the deep communities in how they modify DOM. So now we wanted to look at where are these differences happening in the Van Crevelen diagrams, but they all look the same. And even when we look at the two we want to compare, they still look incredibly similar. So we calculated the relative change over time, and now we have a much clearer picture. In dark blue, it's the molecules that are being consumed after 3 months of incubations. And in warm colors and in red are the molecules that accumulated or have been produced by microbes. And so now we can easily see the differences among the treatments. So in the mesopelagic and bathypelagic treatments in the middle and the right, we see a large dark blue portion on the left side of these graphs. These are the low oxygen molecules that were consumed over time. Now, if we look at the molecules that are being produced, the molecules mostly in red, we see that in the surface treatment, it's all over the place. But as we go down in the ocean, we see these molecules being more and more clustered. So it's more toward the middle for the mesopelagic ocean and toward the right for the bathypelagic over here. And in this region, at about 0.7 to 0.9 in oxygen to carbon ratio, these molecules are considered more recalcitrant. So here we have some clues that the deep communities produce more recalcitrant organic carbon. Now we want to see if these changes in the DOM are reflected in the microbial communities. To do so, we used an MDS. It's an ordination technique to look at the communities. Here, all points are different taxa, and they are colored, again, with their relative change over time. So in dark blue are the taxa that were present at the beginning of the incubations and that disappeared after 3 months. And in dark red are the taxa that appeared or that at least doubled in abundance. And we can see, we can compare again our 3 different treatments. And I'll bring your attentions to here and here, 2 taxa that belongs to the verrucomicrobia that appeared in the deep treatments that are absent in the surface. So again, as with DOM, we see differences in the microbial communities between the surface and the deep ocean. But we can go a bit further than this. We can see if the microbes had a consistent adaptation with DOM. So here we still have an NMDS, but instead of showing the taxa, we show the whole communities as a single point. So in green, we have the surface microbes, in light blue, the mesopelagic, and in dark blue, the bathypelagic communities. And all points are ordered chronologically from time 0 to 6 months of incubation. So we do see a coherent evolution of or adaptation of the microbes as DOM get progressively more recalcitrant. And I believe here that it's often a chicken or the egg question. What is going first, DOM or microbes? I think here we have an example of a feedback loop between the two. We get the pool of carbon to start with, and as it gets progressively more transformed, the microbes adapt to it, and then there's this back and forth between the DOM and the microbial communities. But here we have vastly different communities after 6 months than at time 0. But the microbes that we find after 6 months were already there. Like, they didn't simply appear in the bottles. And these are members of the rare biosphere, microbes that represent less than 0.1% of the abundance. And I see these groups of microbes as metabolic potential or pathways to transform organic matter. And in theory, we have that rare microbes may become abundant again when the conditions becomes favorable to them. So let's see if this happens here. On the left, we have the contribution of the rare biosphere to the biodiversity. So only looking at the proportion of taxa that belong to the rare biosphere. And so we see that rare microbes dominate biodiversity. But if we look at their abundance on the right, we see that in the surface ocean, they never truly were important, at most 27, 28% of the relative abundance. But in the deep ocean, they reach up to 70%. And we believe that it's this rise of the rare biosphere, these microbes that add the metabolic potential to transform this organic matter. And that these rare microbes are a key component of why DOM becomes more refractory at depth. And so here we have a schematic of what could happen during the winter convection. We have labile organic carbon at the surface over here that will be entering to the deep ocean. And as it gets down in the ocean, it will be reprocessed over time, and the microbial communities will adapt to this pool of carbon in this feedback loop. I mentioned a bit earlier that we also had a deep mesoscale eddy. And I think these deep mesoscale eddies act a bit like the winter convection in the Labrador Sea, in the sense that they will mix different water masses together and hence increase the likelihood that certain microbial communities face the right molecules so they can further transform these into more recalcitrant compounds. So if you are interested in a similar topic, it's a shameless plug, I have a poster, number 15. It's a follow-up study on this one. It's on the microbial nitrogen pump instead. So here we will look at how microbes can remove reactive nitrogen from the ocean. And it's using the same experimental setup. So the take-home here is, well, deep ocean microbes do produce more stable DOM through succession of rare prokaryotes. So basically the title of the paper. But also, I mentioned at the beginning, there was a second hypothesis, the dilution hypothesis. Well, because all the treatments reach the same DOC concentration, but they had different molecular composition, this suggests that dilution may play a role in carbon sequestration, at least in the surface ocean, because the DOM pool did not show the same signature as the most recalcitrant carbon in the deep ocean. And finally, that deep mesoscale eddies may be important in the microbial carbon pump by increasing the encounter rates between microbes and DOM. So I'd like to acknowledge the work of everyone that was part of my academic career, all my past and current lab mates and supervisors, Vitals that partly financed my PhD work, all my co-authors, all the people that were on the ship, the CCGS Canadian Coast Guard ship Hudson, the crew members, and the scientists at the Bedford Institute of Oceanography, and my current source of funding in McGill. Thank you. So if you have any questions, there are 2 open mics, and I will respond. I have about 10 minutes for questions.
Hi there. Congratulations on the award, the paper, presentation, really fantastic work. Really interesting time series incubation experiment. I'm wondering if you did any flow cytometry or qPCR or have any idea about the overall cell abundances or any sense of like the transition into dormancy, or if you could talk about that.
Yes, I did use flow cytometry to measure cell abundance, and there were quite differences between the surface and the deep ocean. The surface microbes responded almost immediately, reaching abundances close to bloom. But in the deep ocean, there was, I think, a 7 days or 15 days lag before they took over. And we also measured bacterial production using leucine incorporation. And what's interesting is that when we pooled both information together to get a specific growth rate, we had the reverse, where the deep ocean were the most active cells compared to the surface ocean. And we had this exponential growth and then decrease. So yes, I would assume that part of the microbes went dormant again during the incubations.
Great, thank you so much, and congrats.
Is the really interesting talk as you begin. Is the decline perhaps due to the fact of the microbes being consumed? Because you're talking all about the bottom up. Was there any top down in those experiments?
It's possible. The inoculum was 53 microns meshed, so there could be grazers in there. But we did not measure anything bigger than bacteria. So I cannot say if it's bottom up or top down, or if top down is also at play here. But it's possible because they're in the right size. But I don't have any data on this.
Yeah.
All right.